Hello everyone, welcome to SIUK India webinar. Thank you for joining us today. We are now live with Manchester University. Today we have Professor Sam with us who will let us know more about this special webinar session. Please feel free to post all the questions in the chat box on the right. We will answer all the questions after the presentation during the Q&A session. So let's start. Over to you, Professor Sam.
Thank you very much, Deepika, and thanks everyone for joining. So my name's Sam Griffiths-Jones. I'm a professor in the School of Biological Sciences in the Faculty of Biology, Medicine and Health at the University of Manchester. And one of the things that we're most proud of in the faculty and at the University of Manchester is the really tight link between our teaching and research. And so I have roles in both teaching and research. So I'm a lecturer. But like very many of the teaching staff here in Manchester, I'm an active researcher. And so I have PhD students and I have postdocs in my group, and I direct their research and I perform research myself, and we collaborate with others. But the teaching that I do is driven by this research. And so I'm going to be talking a little bit about my research in this talk, but also a little bit about how it leads into the teaching. So I have those two roles as a lecturer and a researcher, but I actually have a third role which is relevant today too, in that I'm what's called an associate dean, and I'm the associate dean in the Faculty of Biology and Medicine and Health for student marketing, recruitment and admissions. And so I have some academic role and oversight over the admissions processes into all of our undergraduate and our postgraduate master's programs in the faculty. And so I'm mostly going to be talking about my research, but I'll have a little bit at the end about some of the programs that we run in the School of Biological Sciences in the Faculty of Biology, Medicine and Health. And I'm very happy to answer questions about either of those aspects, both about the science and about the courses. Okay, so I'm a professor of computational biology. And so I'm going to be talking a little bit about computational biology. And computational biology is a tool, essentially. And it's a tool that we can apply to all areas of biology, but my group and my research is particularly interested in applying computational biology to the study of RNA, about which I'm going to say plenty in a minute, but also to the study of evolutionary biology and also to the study of development, particularly of animal development. And so I'm going to start by talking a little bit about RNA, which I'm sure you already know plenty about. And what you will probably already know is that RNA acts as a messenger. And so RNA is involved in the transfer of information from our genomes, from our genes, from our DNA. It's involved in the transfer of information from DNA through to making the proteins. The proteins are what we generally think of as the functional bits in cells that are carrying out tasks in cells. And so we have these two processes of transcription of DNA transcribed to make RNA and RNA translated to make proteins. Now, this is, you will have heard about this, I'm sure, in lectures and lessons previously. But we already know that this isn't exactly right. And so there are plenty of RNA molecules that don't act as messengers. And this is a sort of an illustration, I guess, of some of that complexity. So this is an enormous cellular machine, which is called the ribosome. Again, lots of you might recognize this already from the picture. This is a big cellular machine called the ribosome. And the ribosome is actually primarily made up of RNA molecules, not of protein molecules. There are some proteins in the ribosome, of course, but actually the catalytic function of the ribosome, which is to add amino acids onto a growing chain in order to make a protein, that catalytic function is encoded in the RNA molecules that are in the ribosome, not by the protein molecules. Now, what you might be able to see is there's a long kind of brown sort of ribbon over here on the left. There's another long blue ribbon over here, and there's a light green one in the front, which is a little bit difficult to see. And those are all RNA molecules. And as I say, they make up the core of both the structure and the function of the ribosome. These three brightly colored molecules here are tRNAs. So these are transfer RNAs. And see, these are a different type of functional RNA molecule and these tRNAs are showing where they bind in the binding sites in the ribosome. Now, there are, of course, proteins here too. The eagle-eyed amongst you will spot this is an alpha helix down here. So this is clearly a protein. There's some beta sheets up here, some other alpha helices up here. So there are proteins, of course, in this massive cellular machine. But as I say, the ribosome is a ribozyme, which means its enzymatic function is encoded by RNA molecules, not by proteins. And as many of you will know, the ribosome, this is what it does. It's involved in this second step. It's involved in this step of translating messenger RNAs to make proteins. But of course, the ribosome itself has to be made from molecules and largely from RNA molecules that are transcribed from our genomes, from our DNA. And so we know already now then that there are functional RNA molecules that are not just being translated to make proteins, but functioning as RNAs. And so we can say that RNA is not just a messenger. And in the general case, actually, it turns out there are very many of these. I'm going to say a bit about some different classes of these RNAs today. There are very many different RNA molecules. And in the general case, we call them non-coding RNAs or non-protein coding RNAs. But these non-coding RNAs are not translated to make proteins. They function as RNA molecules. Okay, so I'm going to come back to that, but I want to say a little bit more about where I started talking about computational biology at the beginning and why I think computational biology is so important, and bioinformatics is another word for computational biology, and why these studies are so important. And that is because biology, modern biology, 21st century biology, is all about data. It is a very data-driven science. And we can see this immediately if we just start thinking about numbers. We just start thinking about numbers that we come across in biology. And so, for example, we can think about the human genome. The human genome is around about 3 billion bases, 3 billion nucleotides in length. That's a lot of information. It has to be a lot of information because it's all of the information essentially that is encoding a human. Which is obviously massively complicated, but that's a large amount of information and a large amount of data that we can actually use and we can analyze. Now, of course, there's not just one human genome. Every one of us has a genome, has our own human genome. And actually, between any two of us here today, any two people who are listening in, we'd expect there to be around about 3 million differences between any two individuals that are here, unless we've got any relatives, obviously. There'll be fewer differences between siblings. And of course, if we've got any identical twins, then we have essentially 0 differences. But again, that's a lot of information. Just the differences between any two individuals, that's encoding a lot of stuff. There's a lot of things that are determined by those 3 million differences. All of our characteristics, essentially, all of our individual characteristics are encoded by those 3 million differences, very many of our individual characteristics. Now, of course, we are not the only species on Earth, and we are interested in sequencing the species, different genomes of different species on Earth. And we've sequenced tens of thousands of different species. And so we have tens of thousands of genomes. Actually, you could argue we have hundreds of thousands of genomes, depending on how you define whether a genome is finished or not. But tens to hundreds of thousands of genomes of species have had their genomes sequenced. And that sounds like a lot, but actually it's a tiny fraction of the diversity of life on Earth. There are thought to be between 8 and 9 million eukaryotic species on Earth. And so we've actually sampled a relatively small proportion of that diversity. And that's just eukaryotes. It's actually unknown how many bacterial species there are on Earth, but there may be as many as 1 billion, but we don't even know how many bacterial species there are. Now, that's all of those numbers are talking about different species and the genomes of different species. But of course, even within an individual has the same genome in every one of its cells, but how it uses that genome in each of those cells is very different. Different bits of it are turned on and off in different cells at different times. And animals have hundreds of cell types. And so not only do we want to understand all of the information that's within a genome, within a species or an individual, but we also want to understand how that genome is used in hundreds of different cell types in the human body, for example. So huge quantities of data, and we are generating data to understand these kind of issues at an unprecedented rate. The technology available to let us sequence, in particular, genomes has really grown massively over the last few years. And the limiting factor is now not our ability to generate the data, but our ability to analyze the data. And this is why computational biology is so important. As I've said already, 21st century biology is all about data. And I would strongly encourage you, if you haven't already had a chance to try to get some experience of large scale data analysis in biology. So genomes then that we could sequence. This is a sort of historical view of genome sequencing. The first genomes to be sequenced in the 1980s were viruses because they were small, and smaller things are easier to sequence than larger things. And so a couple of hundred megabases, a couple of 100 million nucleotides in the smallest genomes that we sequenced in the 80s. We moved on to sequence bacterial genomes, which are a few megabases, so a few million bases in length. And then into eukaryotes. So Saccharomyces cerevisiae here is a yeast. It's arguably the most important organism on this list because it gives us both bread and beer. But it's the first eukaryote to be sequenced, and it has a genome that's around about 12 megabases in length. And then we get into animals. The first animal to be sequenced was Cerebriditis elegans, which is a worm, which is used as a genetic model up towards 100 megabases. And then the first plant, Arabidopsis thaliana, another genetic model, the fruit fly, Drosophila melanogaster. And then this slide at least culminates in the human genome, which was nominally finished in 2001, of course, although lots of work's still going on, around about 3 billion bases, as I said. And so our ability to sequence these genomes has obviously increased massively, including since the human genome was sequenced. But sequencing itself is not really the thing. What we're really interested in the functional bits of these genomes. And so we can think about how many genes each of these genomes has. And one of the major surprises, of course, I'm talking about protein coding genes first, and I've already told you that protein coding genes are not the whole story, and I'm going to come back to that. But one of the surprises of the Human Genome Project was that the human genome does not have many more genes than some other organisms that we would think of as much simpler. So we may even have fewer protein coding genes than a worm. And that's a bit of a shock to our collective egos. But it turns out that our understanding now says that it's not just about the number of genes that an organism has, but also the complexity with which those genes are used, how they're turned on and off, essentially. And we can measure how these genes are turned on and off using RNA. And so using RNA as a tool. So if we're thinking again about protein coding genes, then I've shown you this scheme where we have transcription of DNA to make RNA and translation of RNA to make protein. And so if we're interested in how turned on a gene is, how a gene is being expressed in a particular time, we can look at how much RNA there is. And we can look at how much RNA there is in a cell, a sample, a tissue, an organism, and we can use we can measure that RNA by sequencing it. And so the same sequencing advances that have let us sequence genomes have also let us sequence the total RNA complement of a sample, which is called its transcriptome. So we can sequence all of the RNAs in a cell, a tissue, a sample, and that tells us which genes are turned on and off in a particular cell. And so this is an example of the power of this. This is quite a complicated slide. It's a slide from a genome browser, a picture from a genome browser. And so this is a little bit of Drosophila melanogaster fruit fly genome that's shown here. And there are 3 genes in this region. There's a gene here called Zen, a gene here called bicoid, and a gene over here called amalgam. And you might have heard of these genes because they're in a very famous and very important region called the Hox complex, which is involved in development. And so there are different isoforms of these genes, so it's already quite complicated. We can see exons and introns of these genes. The introns are the bits that are cut out of genes in eukaryotes, and so they're not involved in making the final messenger RNA that's making the protein. And so below, we've got a bunch of RNA sequencing experiments, so-called RNA-seq experiments. Each line is an RNA-seq experiment. And each of these experiments come from a developmental time course. And where we have a peak, what we're seeing is that we have RNA that has been sequenced from this sample that is mapped back to this region of the genome. So it looks like it is coming from this region of the genome. And so if we have a peak, if we have a bits of RNA that are found to map to this bit of the genome, that tells us that this gene is being transcribed at this particular time. It is turned on, it is being expressed. And so you can see that these genes are turned on and off. All of these genes are expressed at some point, all three of these genes. This gene looks like it's really only expressed very early in development, I should say. This is a developmental time course. So we have embryonic development here, 0 to 2 hours at the top, 2 to 4 hours and so on. You might not be able to read that small writing. And then we have some larval development stages, and then some pupal development stages, and then some adult samples at the bottom. And so this gene over here then is only turned on very early in development, and then it appears to be turned off. Amalgam over here is turned on all the way through, but it looks like it's decreasing in its expression. And this gene bicoid here does something a little bit different. And so it's turned on all the way through embryonic development, and then it's turned off through larval and pupal development, and then it appears to be turned on again in the adult. And this is interesting. And one of the interesting things actually is that this gene, we see lots of transcription from this gene really early in development, even just from 0 to 2 hours of development. And actually at that stage, the embryo hasn't even turned on its own transcription yet. And so the only transcripts, the only RNA molecules in the cells at this point are put there by the mother. They are deposited by the ovary cells of the mother. It is not, the embryo is not yet making its own RNA. And that now tells us something about why it's turned on later, in adults, because actually it's not turned on in all adults, it's turned only on in female adults. And that's because this gene is being expressed in the ovary cells of the mother, so that the ovary cells can put those transcripts into the egg, into the oocytes, because it's needed really early in development. And so we can tell a huge amount about this gene and its importance in development just by looking at when it's expressed, just by sequencing. And of course, there's a huge amount of data analysis involved in this kind of work. This is another view from a genome browser. This now is the human genome, and this is a view from a browser called the UCSC genome browser. This is a couple of hundred kilobases of human chromosome 13. And each of these lines here essentially is a version of an isoform of a gene. So a gene that is being transcribed to make RNA. So here is a collection of isoforms of this gene here, SPRID7. There's another here and another here, another collection here. And you might see that those are slightly darker blue than everything else on this slide. It's a little bit difficult to see on my screen, but hopefully you can see that these three things here are darker blue than the other genes that are shown here. And these dark blue genes are making proteins. They are protein coding genes. And all of the other lighter blue genes here are not. They are making RNA molecules that do not appear to be translated to make proteins. So they are non-coding RNAs. And we know almost nothing about what they do. We know very, very little about what they do. What we do know is this whole region is deleted and downregulated in cohorts of patients who have chronic lymphocytic leukemia. And that leads to the name that you might see of some of these transcripts, some of these genes, DLU, deleted in leukemia. And so we know that these genes are really important in disease, in leukemia, but we don't know what they do. And we think a lot of the disease phenotype that we see comes from these RNA genes, not from these protein coding genes. So we can go back to our table actually and add in RNA genes, and we can see that actually from these sequencing experiments, we see huge numbers of these genes that make RNAs and not proteins. Probably as many, if not more, genes make functional RNAs as make proteins. Now, the differences here are just because of how well studied these organisms are, not because we really expect that worm has nearly 10 times as many RNAs as fly. That's not likely to be a real difference. But where we've done lots of sequencing experiments and found lots of RNAs, we find that there are as many RNA genes as there are protein coding genes in these big complex genomes. And locating and cataloging these genes is essentially a computational problem. Storing the data is a computational problem, and displaying the data is a computational problem. Computational biology has something to say about all of those things. And actually, as soon as I see these numbers, I start thinking about how we're going to do that, how we're going to catalogue these genes and understand them. And there's a quote here from a very famous physicist, Ernest Rutherford, who's a Manchester physicist actually. He was involved in, he determined the nuclear nature of the atom, and he also was involved in splitting the atom in the early 1900s, 1917, I think. And some of that work was done in Manchester. But he said that all science is either physics or stamp collecting. Now, I'm pretty sure he meant this to be a kind of derogatory comment. He was basically trying to say physics is the only important science. But actually, of course, we know that that's not true. But some aspects of biology really are stamp collecting, and proudly so, because cataloging data is really important for our understanding of biology. And actually, a lot of my research is involved in exactly that. And so I'm involved in several resources, online databases, which catalog and help us to understand the function of RNA molecules. And so there's a database called RFAM, which stands for RNA families, and it classifies known RNA molecules into evolutionary families. There's a database called RNA Central, which tries to catalog all non-coding RNAs. So it's a sequence database that catalogs all non-coding RNAs. And these two are both run by the EBI, which is just outside Cambridge, the European Bioinformatics Institute. But I'm involved in both of these. I actually started RFAM when I worked previously at the Sanger Institute before I moved to Manchester. But I also run a database called MIRBase, which is a database of a very specific class of RNAs called microRNAs. And I'm going to talk a bit about those for the rest of the science bit of my talk now. And so microRNAs, again, you might know a little bit about. This is quite a complicated slide, and I won't dwell on it too long. But microRNAs are tiny RNAs. They're only around about 22 nucleotides in length. And on this slide, the microRNA is shown by this little red line here. And what this microRNA does, this tiny RNA does, is it binds to messenger RNAs, so RNAs that are going to encode proteins. These little tiny RNAs bind to messenger RNAs through base pairing. And when they do that, they bring along this big machine, this blue blob here, this big cellular machine, which is called the RNA-induced silencing complex. And the RNA-induced silencing complex shuts down translation of this messenger RNA, and so we're not going to make proteins from this messenger RNA while we have this microRNA bound to it and the RISC bound to it, the RNA-induced silencing complex bound to it. And so microRNAs are a way in which genes can be post-transcriptionally regulated. Most regulation of gene expression happens at the transcriptional level, we think, but microRNAs are a way in which genes can be post-transcriptionally regulated at the level of translation. Now, these microRNAs are made from quite a complicated biogenesis pathway, which I'm not going to go into now, but the important thing to note is that they're expressed from genes that are in the genome. They are transcribed from genes that are in the genome. Now, one of the exciting things about microRNAs is that they're found in all animals, all plants, some viruses. Between 1/2 and 3/4 of all genes in animals are regulated by microRNAs. There are thousands of them in human, a couple of thousand in the human genome, hundreds in genomes like flies and worms, hundreds in plant genomes. We didn't even know they existed before 2001. They are a really recent find. This is really recent biology, and yet they are ubiquitous in multicellular organisms and massively important in regulating gene expression. And that means there's a huge amount we don't know about these things yet because they're so recently discovered. But in the general case, what they do is they downregulate this second step of gene expression, of protein expression from protein coding genes. But of course, we need to remember that these microRNAs are themselves made by transcription. And so they are transcripts that are found in cells. Now, we're interested in lots of aspects of microRNA biology, but including their evolution. And there's another quote here. Nothing in biology makes sense except in the light of evolution. And this is something I strongly believe, that we have to, in order to understand pretty much all aspects of biology, we really have to understand how these sequences and how these functions have evolved. And the core sort of concept here is that functional sequences, important sequences that do stuff in a given given species, are going to be conserved over evolutionary time, and so they are going to be kept somewhat similar over evolutionary time. They're not going to change very much, whereas sequences that don't do so much and not so important, they are going to drift away. They're going to randomly mutate, and they will drift away from each other. And so there's a whole area of genomics called comparative genomics, which is interested in comparing genomes of related species and understanding what we can tell about biology from the similarities and differences of sequences in different species. And this is really the ripest possible time for the study of comparative genomics, because the sheer number of genomes we have available and the diversity of the genomes we have available. These numbers here are almost certainly underestimates, because every time I make this slide, I go and find the numbers, they've gone up. And that's why I put greater than symbols all the way down the right-hand side. We have hundreds of mammalian genomes, thousands of vertebrate genomes. We have genomes that are very closely related to each other. So we have around about 50 Drosophilid genomes, so genomes of Drosophila, a fruit fly species, essentially, Drosophila melanogaster, and 50, around about 50 others. We have thousands of fungal genomes. Fungal genomes are really interesting because we can see diversity across fungal genomes that suggests that they have been diverging for around about 1 billion years. And so we can see conservation between fungal genomes that goes back around about 1 billion years. And that's a quarter of the age of the Earth. And so we can see sequence relationships that go back to a quarter of the age of the Earth. And we can see what's happened in evolutionary time, that kind of evolutionary time. And again, many hundreds of plants. Now, anytime we're going to compare things, compare sequences, again, this is a computational problem. This is a problem, the core problem for computational biology. If we have two sequences, two DNA sequences here, how can we tell whether they are similar or not? How can we judge whether there are enough similarities between these two sequences for us to believe that these sequences have evolved from a common ancestor, for example? Are they the same gene in different species? Again, it all comes back to the concept of evolution conserves important things over time, and less important things will diverge away from each other over time. So we've applied some of these kind of concepts to these tiny RNAs, these microRNAs. And this is a picture from a paper. I thought I had a reference on here, actually. So feel free to email me if you want me to point you in the direction of the paper that this comes from. But what we did was we looked at how microRNAs evolve over time. These tiny RNAs evolve over time. And so we took all of the RNAs that were in the Drosophila melanogaster, the fruit fly. There were around about 250, I think, at the time. And we asked, in what other species can we find the same microRNA? In what other species is this Drosophila melanogaster microRNA conserved? And so we looked in lots of other species, all the way from other Drosophilids, so very closely related species, all the way out to human down the bottom here, which is very divergent from Drosophila, a long way evolved from Drosophila. And so what we could ask is which microRNAs are conserved in other Drosophilids, for example, but not in insects, not in other arthropods, and not in mammals. And if we have a microRNA that is conserved just in Drosophilids, then we can say that we think it arose around about here, around about 40 million years ago. It was born around about 40 million years ago, that microRNA gene. And prior to that, it didn't exist. Whereas if we have a Drosophila melanogaster microRNA that we think we can find in all of these genomes, all the way out to human, then that suggests that it predates the common ancestor of bilaterian animals, which is around about 800 million years ago. And so we have some microRNAs that are very new in evolutionary terms, and we have some microRNAs that are very old in evolutionary terms. And it's really interesting to think about, okay, are those fundamentally different, those microRNAs? Do they do different things? And one of the things that we can do is we can look at when different microRNAs at different ages are expressed. And so this is and we can do this in a couple of different ways. One of the ways we can do this is through fluorescence microscopy, where we can actually attach a fluorescent tag to a probe that identifies when a gene is turned on, when a transcript is made. And so these are Drosophila embryos, two different Drosophila species, Drosophila melanogaster and Drosophila virilis, in a developmental time course from the beginning of development out to a few hours, 10 hours of development. The blue dots show where the nuclei are, and the red dots are showing where a microRNA, this particular microRNA, is turned on. And we can see that it's turned on very early in development when there aren't very many cells, and then we get lots more cells, and they're expressing this microRNA. You see the red dots. And then later on, we start to see that the expression is turned off at the poles of the embryo, and then it's turned off everywhere. And so again, we can say something about this microRNA being important for embryonic development, for early development. We can also look at sequencing data again. And so we can sequence these tiny RNAs again, these microRNAs, and that will tell us, again, if we sequence them, that tells us that those microRNAs have been turned on in that sample. And so this is a set of data that comes from a sequencing experiment that I'll talk you through briefly now. And this is actually from a beetle called Tribolium castaneum. It's around about 300 million years, something like 300 million years diverged from, separated from Drosophila, from fruit fly. But it's still an insect, and so there's lots of common features. And so what we have here is we have lots of different microRNAs, and we have a developmental time course. And so each of these rows then is a sequencing experiment from a developmental time. And Tribolium develops more slowly than Drosophila, and so these are slightly larger numbers than were on a slide earlier for Drosophila. And so the darker the color, then the more highly expressed, turned on, that microRNA is at that developmental time. And what you can see is that lots of microRNAs are expressed late in development, but some are expressed very early in development. And again, we can say something interesting about the function of these things during development by looking at this. Well, one interesting thing that we found is that these are the microRNAs. This is a plot of the microRNAs that are well conserved in other species, and so these are relatively old microRNAs in evolutionary terms. If we look at the new microRNAs, the young microRNAs that tend to be quite species specific, they're not found elsewhere, we see something quite different. What we see is that the young microRNAs appear to be turned on at a very specific time point. They're turned on at this essentially between 5 and 8 hours, and during the 8 to 16 hour stage, these microRNAs are turned on. So we see lots of these young microRNAs, evolutionarily young microRNAs, are turned on at this stage. And this is a really interesting stage, which I've mentioned to you already, because between 5 and 8 hours is the time that Tribolium turns on its own gene transcription. So prior to that, again, the only transcripts that are found in the cell are put there by the mother. And so these microRNAs that are found in these very early stages are actually loaded into the egg by the mother. They're not made by the embryo itself. And then what it looks like happens is after the onset of what's called zygotic transcription, when the zygote turns on its own transcription, after the onset of zygotic transcription, there are a load of microRNAs that are made. And what we find is that these microRNAs are involved in shutting down the other transcripts that were loaded into the egg by the mother, because their job has been done. So once zygotic transcription starts, the maternal transcripts that were loaded by the mother, their job is done, and these microRNAs are all turned on to shut those down. And so in the very early embryo, there's no transcription going on. We only have messenger RNAs and other RNAs that are put there by the mother, but they can be translated to make proteins when we need those proteins in very early development. And then there's this thing called the maternal zygotic transition, when the embryo transitions from maternal RNAs to zygotic RNAs, when the zygote has turned on its own transcription. And now we are having transcription is happening freely in the embryo. But one of the first things that's transcribed are these microRNAs, which shut down these maternally loaded messenger RNAs, so that we don't make these proteins anymore from these maternally loaded messenger RNAs. So this is really quite a cool sort of biological role for these microRNAs, and actually it's now been seen quite widely. Fly, frog, zebrafish, chicken have all been shown to have lots of microRNAs that are expressed basically at the first point at which the embryo is doing its own transcription to shut down these maternally loaded transcripts. Okay, That's just one function, though, of microRNAs, one example of function. But because microRNAs are involved in the regulation of the expression of nearly all genes, then they're essentially fundamental to all biological function. We have loads that are involved in development, loads that are involved in disease. And as I said before, they were unknown before 2001. So really exciting new science. So I'm going to sum up the science bit of the talk, and then I just have a couple of slides on courses. And so I said 21st century biology is really data driven. If you haven't had the opportunity to get involved in some big data analysis, you should try to find an opportunity to do exactly that. Computational biology is really important in modern biology, and it lets us ask and answer really cool biological questions by analyzing data. In terms of the biology of what I've talked about, you should remember that genes don't all make proteins. There are plenty of genes that make functional RNA molecules, and those RNA molecules can be really important, including ribosomal RNAs, tRNAs, and microRNAs are the three classes I've talked about, but there are many, many, many others. These microRNAs are really important regulators of gene expression, as I've said, in development, in disease, and in all sorts of other contexts. But I showed you a specific example of how they're involved in development at this point called the maternal zygotic transition. Okay, I'm very happy to answer some questions on, I can see there are some questions that have been coming in, and so I'm very happy to answer some questions on science, but I'm also going to talk just very briefly about some courses, and you can tell that I'm transitioning here from science to a bit of sort of marketing, because I moved from slides with a boring white background to slides that look a little bit prettier. And so I just want to say just a little bit about some of our master's programs in the biosciences in Manchester. And in particular, these programs, if you're interested in the kind of thing that I've talked about, then these programs might be of interest to you. I said right at the beginning that lots of the teaching that's done in Manchester, and particularly in the biosciences, is really heavily driven by the research that we do. World-leading researchers in Manchester are teaching on these programs, and we teach on our expertise. And so actually I teach on my expertise. I teach on genomics, I teach on transcriptomics, I teach on RNA function. I do a lot of that teaching on the masters in bioinformatics and systems biology. I sometimes also supervise project students on some of these other programs that are related, in particular genomic medicine. But these are probably programs that you might be interested in going to look at if you're interested in the kinds of things that I'm talking about. But of course, as I've tried to say, these kind of data analysis and big data is really fundamental to all biology. And so if you're interested in some other areas of biology that we have master's programs, you will get to touch on these kind of issues anyway. And so we have master's programs in these areas in the life sciences, and computational biology and bioinformatics are relevant to all of these sort of basic biology subjects that are shown here. We also have some much more clinically applied programs, and again they're listed here. And again, it is really hard to do any work in biology these days, in any aspect of biology, without having to consider these kind of big data questions, and therefore be a little bit involved in some computational work. Okay, so I'm not actually going to say any more specifically about courses right now, primarily because actually, if you want detail on what's the difference between any particular two of these programs, then really you want to go and look at our website. And our website for the taught masters programs is at the bottom here. So please go and look at this website if you're interested in these masters programs. If you then have any questions about those programs, then the email address, you'd find the email address on the website, of course, but it's shown here too. And then these guys will be extremely happy to talk to you about aspects of our program, of any of our programs. But I can see that there have been some questions coming through that has been copying across from YouTube into the chat here, which I can start to answer right now. And so please keep those questions coming if you want to do so. My colleague Dan here is from the international office, and he will also be able to help with some questions, I'm sure, that we have here. And so, okay. The first question we have here is from someone: I'd like to pursue a master's in biology as a graduate from Delhi University with 52% and 10 years of working experience. and so I guess the question there is are you able to apply for our master's programs? And so I guess Dan will have an insight into this too. But I think I mean ultimately any question of can I apply is best actually directed at the email address on this slide with the very specific examples of your situation. And so I wouldn't want to give you specific advice based on a very brief summary of your situation, and then for you to find out later that when you actually talk to the programs teams, to the admissions teams, that's not quite right. So I would strongly encourage you to email the address on the bottom here to actually and have them go through all of your history with you. But I would have thought that I would. It sounds to me like we would certainly be interested in talking to you about our master's programs. Dan, do you have anything to add that you'd want to add on that?
Yeah, okay, thanks very much, Sam. So yeah, I'm Dan from the University International Office. So I'm the university's India country manager based here in Manchester. I work very closely with my colleague Aprajita Kalra. So she's based actually in Gurgaon in India. If you want to connect with either of us, then you can as an alternative point of contact. Our email address is india@manchester.ac.uk. And to answer the question around entry requirements in very general terms, we typically look for the Indian equivalent of what we call an upper second in the UK for master's study in a relevant background. So that normally equates to something around 60 to 70% minimum overall in your bachelor's from India. So that's a first class degree in India. So in terms of the 52% from Delhi University, it's unlikely that the admissions team would encourage you to apply. However, as Sam said, I think it's much better for you to email the admissions team and double check on that and how much they factor in your work experience. So hopefully that helps. And as I said, generally for our master's programs in the biosciences, we are looking for you to have a first degree in biological sciences or medical sciences. And then, as I said earlier, your overall percentage should be 60 to 70% minimum normally, depending on which university you went to. For some, it might be slightly higher again. It very much depends. So yeah, case by case, I guess is a short way of summing that up.
And actually, the next couple of questions I can see, I think, are going to be kind of similar in the reason why. So the next two questions actually. So the next one is from a doctor who has nearly 6 years of experience and is looking to do developmental biology. And so again, at the point at which you have lots of extra experience, it really is worth talking to the admissions teams because different courses might weight that experience differently against your qualifications. And so it's very difficult for us to say. To give sort of general guidance when you've also got other things in addition to an undergraduate degree, and so as I say, I think that's going to apply to the next two questions. A doctor with 6 years of experience looking for developmental biology, and the next one has 5 years experience in nursing. Can I apply for this course? If yes, please explain. And so again, we really need you to drop an email to either to the postgraduate talks address on here or the address that Dan just read out, which I'm sorry I didn't have on my slide, to start a conversation about the other things that you have that might compensate if you don't quite meet the requirements that Dan's talking about for an undergraduate. And actually, Dan, there's another general point here. If people are have qualifications that they've done a long time ago, which is also going to apply for the fourth question here, which is somebody who completed MBBS in 2015, can I apply for masters for September 2022 intake? And so, do you know anything about about issues that students might come across if they've had a gap in their studies? So if they've done an undergraduate qualification a long time ago, whether they might need to do anything else before they can come in to do a master's with us.
There isn't a requirement to have done anything else as such, but I think the admissions team will be interested in what you have done in the interim when they're assessing your application. So if you've been working in a relevant field, for example, I think that will be well viewed. But it is a bit case by case there. So again, I'd probably recommend that you email the admissions team to double check. I would suggest that you attach your transcripts of results from your degree, and then your CV alongside that, and ask them whether they'd encourage you to apply or not.
Okay, good, thank you. The next question is about scholarships. So for scholarships, can I apply separately, or is it considered automatically when you apply via UCAS? So there's already a little bit of a wrinkle here, because of course, UCAS, the courses I mentioned here are master's programs. And so you don't apply through UCAS for master's programs. And so if you're thinking about applying through UCAS, then perhaps you're thinking of applying for undergraduate programs, which is a different system. So if you're looking to come and do an undergraduate degree in the UK, then UCAS is the correct application route. If you're looking to come and do a master's degree, then you will apply directly to the universities that you are interested in, including hopefully us. But Dan, can you say a bit more about scholarships?
Yeah, absolutely. So at Manchester, we do have our Global Future Scholarship Scheme for undergraduates across the institution. And in order to apply for the scholarship, you would need to have an offer from Manchester, and then we would email you with the scholarship application form. You'd find out if you were successful or not at some point between June and July in the year that you're coming to Manchester. So the value of the scholarship is £5,000. So it's not a full scholarship, it's a partial scholarship, but it's at least something that you'll be able to apply for in that regard.
Okay, good, thank you. The next question is, what are the top UK employers post completing a biology program? Okay, so I have sort of two answers to this. One is I can give you some stats. Actually, the stats I have in my head are what our undergraduates go on to do. Dan might have something also about some postgraduates, but I have some stats in my head about what our undergraduates go on to do, and I can imagine that it's actually fairly similar across the PGT courses. But actually, I'm going to start by saying I don't think you should care about this. Now, you might find that a little unusual, but the reason I'm saying that is that I'm very much an advocate that you do the thing that you are really interested in doing. That if you're interested in a master's program, then you do it because it's the thing you really want to do and you really love, and because if that's the thing, if that's true, you really enjoy something and you really want to do it, then you are going to do better at it, and then you're going to get a better grade. And at that point, there's a huge quantity of careers that open up to you. And so that might sound a little bit odd, but I'm going to now reassure you in that view by saying that if you were to come to Manchester to do either an undergraduate or a master's, actually, then you will be in very good stead for a very wide range of careers. And so, as I say, the stats I have in my head are for what our undergraduates go and do, which is that around about 2/3 of them go into some career that is directly related to science. And about half of that, so about a third of the total, go on to do further studies. So many PhDs, for example, and that's a very strong route for master's qualifications to progress on to too. And the other third go and do something else that is not directly using the biological knowledge that they have, but is very much using the problem-solving knowledge that they have. Because actually what a degree and a master's and further a PhD teach you, in my view, the biggest thing that they teach you if you work in the biosciences is problem-solving skills. And the really great thing about problem-solving skills is that they're applicable across a huge range of careers. And so there's, as I say, around about a third of our undergraduates, I imagine it's a bit smaller actually for PGT, for master's graduates, but around about a third of our undergraduates go and do something that is not directly related to the science, but is very much directly related to their problem-solving ability. Unfortunately, those people often go and earn more money than the people who are staying in the science programs, in going on to do further science. But of course, lots of us do science because it's the thing we love. But I strongly encourage you to find the thing that you are really interested in doing and do it well. And at that point, a huge range of careers will open up to you. The other thing I want to say there is that Manchester is a really excellent place to be for career prospects in general. And there are two sort of strong indications of that I can give you. One is that our careers service in Manchester is consistently voted the best university careers service in the country. I have a slide that I use in an undergraduate talk that I've used in an undergraduate talk for the last 9 years. And every year, all I've had to do is change the date of that award, because every year the University of Manchester's Careers Service has been voted the best in the country by employers. That's both by students and employers, actually. But the other is that Manchester has for a while been the highest rated university in the UK by graduate employers, by the top 100 graduate employers, and so you can be sure that a degree or a master's or both from the University of Manchester will stand you in extremely good stead for careers. But as I say, I think you should focus on finding the thing you really want to do at the time you want to do it, rather than worrying about too much about where it's going. Again, Dan, do you have anything to add to?
Yeah, I thought that was fantastic advice for anybody listening, really. So yeah, what you get at Manchester really is fantastic access to leading employers. So they're regularly on our campus giving presentations, there are careers fairs, etc. And you get excellent support from our careers service as well. That's not to say that it isn't competitive here. It's competitive to get those jobs, but yeah, you do have good access, good opportunities, good support. So I always use the gym analogy. If you're coming to a university in the UK like Manchester, it's like you're paying for an expensive gym, and then you need to get here and kind of use the weights and the running machines and so on to get yourself on track. And in terms of specifically where people go on to work from this kind of background, it's quite often, I think, as far as I know, correct me if I'm wrong, Sam, but is it like pharmaceutical companies, medical research sometimes? Is that fair to say? Perhaps health data, biotech?
Yeah. Actually, I was talking. I guess I was talking fairly broadly about the biosciences in general. But yeah, no, for the kind of things I've talked about in terms of sort of big biology, data science, then yes, absolutely, lots and lots of routes into pharmaceutical companies and biotech. But actually, again, plenty of routes outside of biology if you decide at that point that you don't want to remain in the science, because the ability to analyze data is crucial across huge swathes of industries, enormous ranges of industries. And so data science is arguably one of the biggest growing industries in the world across a whole range of fields, unsurprisingly, of course. Okay, there's a couple of more practical questions here. I'm sorry, Dan, these might also be for you. Is there new COVID guidance for Indian students, quarantine or anything? Will the university assist us with quarantine and things like that? I guess is that question.
Yeah, I guess to start with, I can say that on a general note, we're obviously very supportive of our students from India, and we'll do everything we can to kind of welcome you and accommodate you. In terms of COVID and the guidance, as things stand, you wouldn't need to quarantine on arrival, assuming you can prove that you are double vaccinated. So you would arrive and you'd need to do a test on arrival to check whether you have COVID or not. Assuming that you've managed to travel and not contract COVID, there wouldn't be any kind of form of quarantine arrangement, anything like that. So you'd be able to participate in activity on campus relatively normally as things stand. We do advise social distancing and wearing masks in shared spaces and so on. So we are being careful, but we've kind of resumed relative normality really to a large degree in terms of our teaching here at Manchester. So that's a bit of a short answer. There's a lot more I could tell you there. So if you're interested, then I'll just repeat the email address I gave earlier, which is india@manchester.ac.uk. Any general questions like that, feel free to get in touch and we'll be happy to help you. The next one I think is around accommodation. Yes, we have accommodation here on our campus, as you can see behind me. So it's a campus inside the city centre. So there's accommodation right in the city centre of Manchester, or within kind of 10 minutes walk. And then we have accommodation 50 minutes walk away in Victoria Park and about 25 minutes walk away in Fallowfield. It's all very well connected, really good transport links. And there's a range of accommodation available. So you can have en-suite if you wish. There's always kind of shared living area, shared kitchen facilities. There are catering services available in some of the accommodation as well. So yeah, huge range. You can find the details on our website. It's just accommodation.manchester.ac.uk. Again, if you want more information about accommodation, just drop me a line, no problem. Discount for prior booking? No, we don't offer that. But I can tell you that you can apply for the accommodation as soon as you receive your offer. And it's a good idea to do so. But yeah, more questions about accommodation, please drop me a line.
Good, thank you. There's then a couple more questions I can see about people's specific circumstances. So there's one here about someone who's completed a bachelor's in something completely different, in fashion design. So can I apply for this course? Now, usually we would require that you have done some kind of biological science as an undergraduate. If you think there's something else that you have that might mean that you can meet those requirements in some other way, then again, that's the right time to email the admissions teams and talk to the admissions teams directly about your background, rather than have us try to infer an answer from a very short description of your background. And so please get in contact with the admissions teams for that. The next question is, do we need to submit a personal statement along with our application? And so the application is via an application form that you'll find the system for on the university website. And as part of that, there will be an opportunity for you to describe your interest in the programs, and you should definitely take advantage of that opportunity and do exactly that. Talk about why this is the program that you are really interested in, experience you have that's directly relevant to that, even what you might be thinking of doing afterwards. So you should certainly take the opportunity to explain why you are interested in a particular program, because the admissions tutors will definitely be interested in that. And what are the job prospects for this course, which I've said a little bit about already. And then there's another specific circumstances question. So I have a 2-year gap after my undergraduate. Can I still apply? Again, I'm afraid that it will depend very much on your personal circumstances. So that in itself won't mean that you can't apply, but you really need to get in contact with the admissions office. Please don't be shy about doing that. They want to hear from you. The admissions teams want to hear from you. We want to talk to you about your circumstances. We want to help you put in an application that is appropriate to your circumstances. So please don't be shy and get in contact with that email address. What are the career opportunities? I think I've said a little bit about that, so I'll skip over that one. Someone who's currently doing an MSc in zoology but is planning to apply for an MSc in cognitive and clinical neuroscience. And so am I eligible for this course? So certainly the MSc in zoology won't, that doesn't make you ineligible for the course, but your eligibility will be determined by some other factors too. So again, please get in contact with the admissions team. And I think the last question I can see, I've applied for my master's in bioinformatics. My application has been under consideration for 3 months now. When is the earliest I get to know about it? Okay, so there's a general answer to that question. I have a more specific answer in a minute, but a general answer to that question. If you think you're not hearing in the timescale that you should be, drop the office an email. They'll be perfectly happy to tell you what's going on. It could be that the application is being considered by a program director, for example. It could be that we're waiting for something from you and you've missed an email saying that we need a particular document or something. And so if it's taking longer than you think it should be, get in contact and we'll find out why. Now, for the bioinformatics one, I happen to have some information which might be why, and that is that the bioinformatics program operates a staged admissions process. And that means that it considers admission applications that come in a particular time frame, and it considers all of those first. And then the people who've applied in the next time frame are sort of put on hold while this first time frame is considered. Now, it can be a little bit confusing, but actually getting into the time frame and being eligible for the first time frame, for example, requires that your application is complete at that point. And so if, again, we're waiting for something from you, evidence of an English language qualification, for example, if we're waiting for that from you, that means your application is not yet complete and it won't have been flagged as now being ready to be examined by the program directors. And so it could be, again, that you think you're waiting for us, but actually we think we're waiting for you. And so if there's any confusion there, the best way to resolve that is to get in contact, email the office, and they will be very happy to talk to you about why that is. So I hope that makes sense. I think that's all the questions I can see. Pika, is that everything?
Yes, I think we have covered all the questions. And I would say thank you so much for answering all the questions so well and sharing your knowledge with us. We are now moving towards the end of this seminar. I would request if you have anything else you would like to put forward for the audience who are watching us live right now.
No, I don't think I have anything else to say. I've said it several times already, but I'm going to say it once more just to reinforce it. If any questions at all, anything, then you should email us. And so Dan's given you the email india@manchester.ac.uk if you've got questions that you think might be specific to the fact that you're applying from India. If you have questions about admissions criteria or the state of your application, then the email address on this page is the correct one. So pgtalks.biosciences@manchester.ac.uk. If you want to talk to me about science, then my email address is there too.
Thank you so much, Dan and Sam, for your kind words. To all the audience, if you have any further questions, please feel free to contact our SIUK India offices. We'll be more than happy to help you. And in the end, I would say thank you so much, Dan and Sam, for your valuable time today. So we are wrapping up this session. Thank you. Bye-bye.